Hello, everyone. Good afternoon. Today, our MGFA research webinar series will be our argenx clinical trial update, Adapt Seron with Dr. Guptill. Dr. Guptill is the neuromuscular franchise lead in clinical development at argenx. In this role, he oversees the late-stage clinical development programs for efgartigimod and EMPA. I'm told I can say the short word. He is also adjunct associate professor of neurology at Duke University School of Medicine. Dr. Guptill received his medical training at Virginia Commonwealth University and subsequently completed an adult neurology residency in neuromuscular medicine, EMG, and advanced neuromuscular disease fellowships at Duke University. Prior to joining argenx, he was a tenured professor of neurology at Duke. We also do want to sorry about that. We want to thank our sponsors today, our national presenting sponsors, argenx and Alexion, our national collaborator sponsor, Amgen, and our national supporters, UCB and Johnson & Johnson. I am going to stop sharing. I'm going to let Dr. Guptill share his slides and begin the presentation.
Okay, and you can see the slides, Tova?
Yes, perfect.
Okay, great. All right, well, thank you, Dova, for the introduction, and thanks for everyone who's attending the this webinar today. As Dova mentioned, I'm going to be talking about a study that argenx is planning to do in patients with AChR negative generalized myasthenia gravis, and it's called Adapt Seron. And right up front, I'll start by saying the patient population for this study includes basically all patients with generalized myasthenia gravis who don't have AChR antibodies or the acetylcholine receptor autoantibodies. That tends to be an immediate question that people have, and so I just wanted to address that up front. So it's a pretty broad population of patients, and we'll talk about this in more detail as we move along. So I'll give some information first on what clinical studies are for those of the audience that aren't familiar with them and why they're conducted. We'll talk about the seronegative myasthenia gravis patient population, and then I'll talk about the study itself and give you some information about where to turn to if you're potentially interested in learning more about it. And then at the end, we'll have some questions, as Dova mentioned. So first, we'll start with what a clinical study is. So the clinical study is an experiment, basically, to learn whether a investigational study drug is safe and effective for treatment of patients with certain diseases. And these studies are generally conducted in a very rigorous fashion with very close observation and follow-up and very high standards for how data is collected and analyzed. And that's for a number of purposes, but including to ensure that the study drug is safe and effective, and also to ensure that that patients who may participate in the study are being treated well and fairly, and that their best interests are taken into account and are safe as much as possible during the conduct of the study. So there's a protocol that lays out all the different parameters of the study, and that includes the types of patients who are eligible to participate, the schedule of the dates when participants would need to come in to have the testing and procedures done, what the study drug is and how much they're given and in what way it's administered and how long the study is, as well as the overall objectives and endpoints for the trial itself. So it's a very controlled process. And I think that's important to know because I was formerly an investigator at a clinical trial site in the past, and there were patients who were interested in participating in a particular clinical study, but didn't meet the criteria for it, and sometimes that can be frustrating. But it's done in a way to ensure that that we can get the result of the study appropriately to understand the effect of the drug in terms of how well it can help patients potentially, but also to ensure that it's safe. So as I mentioned, these studies are critically important to evaluate the safety and efficacy of new treatments before they're made available to the public. And they also help to expand medical knowledge by providing evidence that a particular study drug is effective, and that evidence is collected in a way that's very high quality that people can trust. And then that can therefore then if a drug is approved for a certain group of patients, that information is available to help the patients and their healthcare providers make informed decisions about whether or not a particular treatment is appropriate for them. So in this particular study, the investigational drug is called efgartigimod alpha, or efgartigimod for short. And it is already approved for AChR positive, antibody positive, generalized myasthenia gravis, and is being evaluated in a number of other severe autoimmune diseases. And we'll talk about how efgartigimod works on subsequent slide. But efgartigimod is not approved currently for the treatment of AChR antibody seronegative GMG, and so that's why we're performing the study is to ensure that we have enough high quality evidence to say that it works for the majority or the overall population compared to their current treatments that they may be receiving. And so I will actually take a moment here to speak a little bit more about what efgartigimod does. So efgartigimod lowers IgG, and so some of you may know that autoantibodies, including AChR antibodies, MuSK antibodies, LRP4 antibodies, are IgG molecules, and these autoantibodies attack the muscle. And so efgartigimod works by lowering IgG levels, including the autoantibodies. And the idea is that if we can remove these autoantibodies from the circulation, that the clinical status of patients will improve, that they'll regain function and their symptoms will get better. So what is AChR antibody seronegative myasthenia? So some of you may know this already, but we'll just give a quick background for those of you who aren't familiar with it. So in general, autoimmune myasthenia gravis occurs when the patient's own immune system attacks the muscle or the neuromuscular junction and the communication between the nerve and the muscle. And so this is, broadly speaking, this is classified as a neuromuscular junction disorder. So, and it's autoimmune in nature. And the result of the disruption between the communication of the nerve, the motor nerve, and the muscle is weakness and fatigue, which can be quite debilitating for patients and even life-threatening in some circumstances. Among the overall population of myasthenia gravis patients, there is a subgroup of patients, about 20% of them, that don't have the AChR autoantibodies. And so 80% or 80 to 85% of patients do have AChR antibodies that you can detect on a blood test. And that is the population that has been studied the most for myasthenia. And so what we're really interested in is to gain more information on how patients, the subgroup of patients that don't have AChR antibodies, responds to efgartigimod. And again, I'll emphasize several times throughout the talk that this is for patients that have MuSK antibodies, those that may have LRP4 antibodies, as well as patients with no identifiable autoantibodies, which sometimes are called the triple or double seronegative myasthenia gravis patients. So currently, this subgroup of patients, the seronegative group of patients, they of course are receiving treatments, the treatments that are typically used for AChR positive GMG in a lot of cases. But specifically for the subgroup of patients, these treatments have not been really studied in rigorous clinical trials before. So this is a unique opportunity for us to learn more about efgartigimod in a very high quality, large study to inform whether or not this treatment may be appropriate for seronegative patients. So now we'll turn to the study itself. The study is called ADAPT Seron, and the Seron short for seronegative. And this is a global clinical trial designed for patients with seronegative generalized myasthenia. And it will include about 22 countries, and including the United States, of course. And we're anticipating about 18 sites from across the United States. And the study is designed to evaluate the efficacy, how well it works, as well as the safety of efgartigimod compared to placebo, meaning like a water infusion as a treatment. And I'll tell you more in a couple slides about the study design, and I think that'll make it clear for people. And now just generally, I'll talk about how long the study is. So the study itself will last for up to 2 years, approximately 2 years. And during this time, participants will need to come to the site that they're doing the trial at defined intervals to receive treatments with the study drug, as well as to perform other assessments that are part of the study protocol that I mentioned before. An important part of that is also to make sure that if patients are having any adverse effects or having any problems with the medication, that those are documented and managed appropriately to ensure that participants are safe. Within the overall study, there are several parts. So and we typically break these up. I'll show a schematic in a couple slides that I think will make it a little bit clearer. But in general, there's a period where we look to see whether or not someone who wants to participate in the study is eligible for it. So there's some testing that needs to be done during what's called a screening period. And that includes like an EKG, so a heart rhythm test, includes blood testing, as well as some clinical assessments of their severity of their myasthenia. And then there's also some information that needs to be submitted about how a patient was diagnosed with seronegative myasthenia. In some cases, because some patients may not have any autoantibodies identified in their blood, it's important to really be sure that they, in fact, have the correct diagnosis and that they truly have myasthenia gravis. So part of the study also includes a committee that will review data that's being submitted as part of the trial to review the diagnosis of myasthenia gravis. The next part of the study, if a participant is eligible, they'll then enter what's called Part A. And so Part A of the study consists of a 3-week treatment period and a 5-week follow-up period. And this part of the study is double-blinded, meaning that no one knows like what treatment the participant is receiving. And there's 2 options that the participant can get, either efgartigimod or the placebo, which is the water infusion. And this is done so that there are no biases among any of the people who are involved in the study including the study staff, to make an assessment of whether the treatment works or not. So no one will know if they're getting the active treatment. And then after Part A, then patients will move to Part B, which lasts for up to 2 years. And during Part B, all the participants will receive efgartigimod. So there's no placebo in that part of the study. So everyone gets efgartigimod. An important thing to note is that a participant who enters the study is free to leave at any point and for any reason. And I think in my past experience, that was also a question that patients had, was whether, if I commit to this study, is there any way out for me, so to speak, if I move, or something happens that I need to withdraw. And so I think that's important to know, is that you can withdraw at any time. So now I'll give you a little bit more detail about the study itself. And I've already mentioned that there are very strict schedule of evaluations that are done as part of the study to monitor the effect and the experience that the participant is having from the study medication. So this is a schematic overview of the study. I've already highlighted several parts of the study already, but we'll go over them again to make sure that they're clear. And we'll work through this slide from left to right, sorry. So on the left-hand side of the slide is the screening period that lasts for up to 5 weeks. And again, this is where we're checking for the eligibility of the participant, that they qualify and meet the requirements to enter the study. If a participant is eligible for the study, they then move to Part A, and you can see that as the next little group in green there. And so in Part A, as I mentioned, it's 3 weeks of treatment. So you get 4 doses of active drug or placebo, and then that's followed by a 5-week follow-up period. At the start of Part A, there's something called randomization, which is essentially like a flip of a coin. And during that coin flip, the participant is either assigned to the treatment arm that'll get active efgartigimod, or to the treatment arm that gets the placebo, the water infusion. And again, no one knows which arm the study participant is in. And again, that's called blinding. And so after that randomization, there's the dosing in Part A, and then the 5-week follow-up period. At the end of the 5-week follow-up period, the participant then moves on to Part B. And as I mentioned, the Part B, everyone receives efgartigimod. So again, no placebo. So the placebo part is relatively short compared to a lot of studies. So it's about 8 weeks of the double-blind period where a participant could be on placebo. And then the rest of the time, approximately 2 years, the participants would be on efgartigimod. And so in this part in Part B, the participants will get cycles of efgartigimod given in infusions in weekly infusions that are given in groups of 4, so 4 weekly infusions. And then this is followed up by a follow-up period. At the beginning of Part B, the follow-up period is fixed, meaning that the time between the next cycle is predefined. But then after that, as the study progresses, the participants' treatment can be adjusted based on their clinical response. And we know from prior studies in the AChR positive GMG population that some patients, need more or less of efgartigimod to manage their treatment. So this we think is a nice element of the design, in the sense that it takes into account the individual needs of the patient as they progress through the study. And it's not a one-size-fits-all for all patients. The primary endpoint for the study is the MG-ADL score at the end of the double-blind period. And some of you may be familiar with the MG-ADL already from your clinic experience, and I'll talk about that more in a little bit. So I've talked about the different parts of the study. So we have part A and part B, but there are also two different types of visits that can occur. There are treatment visits where you get the investigational treatment, you get the study drug or placebo if you're in part A, or in part B, you just get efgartigimod. And then you also have other follow-up visits. And in the follow-up visits is where we're doing additional monitoring to see to ensure that patients are being monitored appropriately for safety reasons, as well as to measure the effect of the treatment on their MG. The infusions themselves last about an hour, and then there's a 30-minute observation period after that. Of course, the study visits themselves last longer than that because there are other assessments that need to be performed during that time. But the actual infusion of the study medication is an hour. So, and I've alluded to this already, but the purpose of the trial is to look at how efgartigimod compares to placebo in patients who are AChR seronegative and have a generalized myasthenia gravis. One other thing that I didn't mention earlier is that patients can come into the study on existing treatments. And so we're essentially looking at how efgartigimod does in addition to any treatment that they're already taking. And during the trial, at least the early stages of the trial, the existing treatments that patients are taking must remain stable. And that's intended to ensure that if there's a change in the patient's status, say, for instance, the patient gets better, we'll be able to know, is it because of the efgartigimod treatment, whereas if they get better and they'd had one of their other treatments that they were on adjusted, we won't know which is which. So that's why it's important for it to stay stable throughout the trial. I've spoken about this already, but I think it's important to emphasize that we need to do this study for multiple reasons in our viewpoint. So one is that we don't have a lot of information from high quality, rigorously conducted studies about treatments for seronegative MG. And so we're hoping to partially fill that gap with this particular trial. And as a result of the exclusion from prior clinical trials, we feel that there's still a high unmet need for this subgroup of patients. And again, we're trying to evaluate whether efgartigimod can be an appropriate treatment for these patients. And ultimately, this would hopefully lead to, if it's successful, to an approval through the FDA for efgartigimod in this subgroup of patients. And this step is important because for a couple of reasons. One is it ensures that there's a review by a regulatory agency to say that the trial was conducted appropriately and the results have been interpreted correctly. And it also provides access to patients for the particular treatment. So overall, we're looking to include about 110 patients. And again, I'll emphasize they can be MuSK antibody positive, they can be LRP4 positive, or they can have no identifiable autoantibodies. And we're looking to enroll as diverse a population as possible. We need to learn as much as we can about this treatment and to see if there's any differences in the effects based on patient characteristics. And that's one of the reasons why we also have a global study. So we have a global study where we're enrolling patients from many different countries and regions of the world to study the effects broadly. So how will the efficacy of the drug, or how well it works, how will that be evaluated? So in the most critical part of the study will be in part A, and that's when we're comparing during the blinded period when no one knows what treatment they're on. And so in that period, we can hopefully in an unbiased way look at whether efgartigimod does more than providing just a water infusion. And so this will be done on using two main scales. So one I mentioned already, which is the MG-ADL scale. This has been the primary outcome measure in the majority of clinical trials that have been done for myasthenia gravis, or at least the drug trials. And this scale measures patient symptoms, and you again may be familiar with this from your clinic. Is that some neurologists will perform the ADL score during clinic visits, and then if you've ever participated in, say, the MG registry that MGFA has, or some other studies, the MG-ADL is a very commonly administered scale. And then the second major scale is the QMG, or Quantitative Myasthenia Gravis score. And this is a healthcare administered evaluation of muscle strength and fatigue. And this is also a common measure that's incorporated into clinical trials. So I also wanted to spend some time emphasizing the receiving of study medication. And I know that participant or patients who consider clinical trials, they're concerned in some cases about not being able to receive the study medication. So I want to make sure that this element is clear. So in part A, that's the part where we have the placebo control, and that's where a participant has a 50-50 shot of getting either the water infusion placebo or efgartigimod. And again, this part of the study is relatively short, so it's 8 weeks. And then after that, in part B, it's open label, meaning that everyone gets efgartigimod. So if you're eligible for the study and a participant goes through part A, they would expect to, in part B, all be able to receive efgartigimod, even if they were assigned to the placebo group in part A. So, and this is done in part B to evaluate the long-term safety of efgartigimod in the group, but it also provides an opportunity for all participants in the trial to have access to the study drug, even if they were assigned to placebo in part A. So both of the efgartigimod as well as the placebo are given through an intravenous infusion, and again, this is done so that you can't tell which one is which. Another key element for clinical trials is privacy, and so wanted to spend a minute on this. And so the study teams treat privacy issues very seriously. So the sponsor, meaning argenx, as well as the companies that may help us with the clinical trial, and that goes all the way down to the study doctors and the staff that are at the site. That everyone needs to be handling a participant's personal health information in a confidential manner. And to do this, there's many different elements that are involved, but one important one is to assign a study number to every participant. So names and other identifiable information are not used in the records, names and initials and things like that, but they're given study numbers to help protect confidentiality. And as I said, there are many elements to this, but that's just one important piece. So what else might you want to know? So it's possible, of course, since this is a clinical study, that you may not benefit from the treatment if you receive efgartigimod. It's possible that the symptoms could get better, but it's also possible that they could stay the same or get worse. And so that's something to know in a clinical trial, is that nothing is guaranteed. And that's one of the reasons why we're doing the study. If we knew how people were gonna react, there may not be a need to even do this study. But I think in this case, we have confidence to think that it should work based on our understanding of the biology and our experience with other clinical studies that have been done in myasthenia gravis to date. So, and another aspect that it can, for some patients, be rewarding and a reason to participate is that not just for their own personal benefit, that their own myasthenia gravis may get better. Of course, that's obviously important for a lot of people, but a lot of people also see participation in clinical trials as a way to contribute to the body of information, the body of knowledge that's out there, and that other patients and healthcare providers will be able to use in the future in making treatment decisions. There's no cost to participate in the trial, and there are mechanisms to help cover travel costs and travel related expenses. I know a lot of patients may have to travel long distances to get to clinical trial sites in some cases, and the clinical trial itself will reimburse for those expenses. We don't want those elements to be a barrier to participation in the clinical trial. All of the treatments and tests that are done in the trial are also paid for by the study itself. It's possible that some study visits can take place at home with a study nurse. That would mainly be in Part B, the later end of the study after we get through the Part A, which would be the placebo-controlled portion. And so that's about it. So if you're interested in learning more about the study, you can talk to your treating physician that manages your MG. There is a website that's been set up also to provide some information. You can see it here on the screen, as well as a QR code if you're familiar with that. You can use your phone to and that'll take you to the website to give you some more information about the study. And then finally, as another way to learn more, there's also a phone number that you can call, a hotline, to get more information about the study. So with that, I will close, and I will open it up to see if there's any questions.
We do. Thank you so much, Dr. Guptill. We do have some questions. I think there'll be some more coming in as we go, but I also have some that were sent to me. So the first question is there a reason these patients are not screened for VGCC antibodies to ensure they are not incorrectly categorized as seronegative?
Yeah, so I think the question is referring to voltage-gated calcium channel antibodies, which are diagnostic of a condition called Lambert-Eaton myasthenic syndrome, which is also an autoimmune condition. So typically, Lambert-Eaton myasthenia can be distinguished clinically based on the clinical features of the participant itself or of the patient themselves. Typically, in my experience, there are unique aspects of the clinical presentation as well as the evaluation that will point to the diagnosis of LEMS versus myasthenia gravis. So for that reason, it hasn't been mandated to have a voltage-gated calcium channel antibody test as part of the clinical trial design. Now, if a patient has had that testing done before, that will be taken into account, like in terms of the eligibility of the participant, but it's not required for eligibility.
Would you mind putting the slide back up with the website and the phone number? So if people need to copy it down, they can. Perfect. Okay. I just wanted them to have it in case.
Yeah, no, that's good. Thank you. Yeah.
Is a recent thymectomy part of an exclusion criteria for the study?
Yeah, so there is an exclusion for thymectomy. I believe it's 6 months. Yeah, but if it's beyond that, then it's fine.
Okay. The next question is, will the patient have to travel to the clinical trial site for the extended use portion?
Yeah, so for that, yes, the patients will need to travel to go to the sites for certain visits. There may be certain visits depending on where a participant lives, where they may be able to do like some assessments remotely, and we want to try to provide that for participants, to make it as feasible as possible, and less as as minimize the burden of the trial as much as possible. I mean, I think we certainly recognize that it's quite an investment to participate in a clinical trial, and we don't want to be doing things unnecessarily that we can, when we can get information in other ways.
And the next question is, can you be on all existing treatments, including Soliris?
Including what? Sorry?
Soliris.
Soliris. Okay. Yeah. So there is so in general, you can be on the oral treatments for MG and stable doses. So that would include like pyridostigmine, mestinon, oral steroids, mycophenolate, which is also called Cellcept, and azathioprine, which is Imuran, and then methotrexate and tacrolimus. So those are kind of the most common oral treatments that are used for myasthenia. Those are all acceptable, and again, there are rules in terms of like how long the doses have to be stable for those to meet eligibility. In terms of other classes of drugs, which we have biologics, which include like rituximab and also Soliris, or also known as eculizumab or ravulizumab, which is Ultomiris. We do have an exclusion of that. You can't be on those on the complement inhibitors like eculizumab for 3 months. And there's a specific reason for that, is that also like efgartigimod or sorry, Soliris is an antibody essentially. And so what will happen is if we give efgartigimod to a patient who's on Soliris, we'll remove the Soliris from the system. So there is a washout period, we call it, for that for those medications. For rituximab, the washout is a little bit longer, because we also know that the clinical effect of rituximab for patients who respond tends to be longer.
Okay, great, thank you. The next question is, so many seronegative patients are already getting treatment. Will this impact the results of a repetitive nerve study if it needs to be done for inclusion criteria?
Okay, so I think I understand the question. Yeah, so in most cases, patients have had their neurophysiology tests done at the time that they were diagnosed. Sometimes they'll get them as follow-up tests along the way after they've started treatment. But most of the time, those tests have been done when they've been off of treatment, or maybe just on minimal treatment. And so in some cases, for this study, it may be necessary to repeat the studies, the neurophysiology studies. For instance, if the study was done a really long time ago and the results aren't available, for some patients, it may be necessary to repeat the studies. And in that case, a lot of times the patients will be on treatment at that point in time. And it is true that it's possible that the results of the test may be less likely to be positive if they're getting restudied at a time when they're on treatment. However, I would think that if a patient was considering participating in the trial and had symptoms that were severe enough to warrant being in the trial, that the repetitive nerve stimulation study or single fiber EMG study should still be positive, should still give the answer that it's consistent with MG. The single fiber EMG test, it would be more sensitive for detecting the evidence of the abnormality than repetitive nerve stimulation. But of course, single fiber EMG isn't available everywhere. It's a more restricted test.
Okay, thank you. The next question is, have test sites been determined? And if so, is there a list available?
Have the sites been
Have the
determined?
sites been determined?
Yes, so so not all of the sites, but there are some of the sites have been determined. I'm not going to try to like list the sites, but so clinicaltrials.gov can give information like on the availability of clinical sites. And then also on the website and that link that I provided, if you go in there and kind of register, and you can put in like where you live, it can help you to link with a potential site that may be close to you.
Great. The next question I think is asking, will you be taking a certain number of the different types of MG patients? So you said seronegative must. Is there a certain number for each type that you'll be accepting into the trial? Or is it first come?
Yeah, so at this point, it's whoever first come, first serve is the way that we're doing it. Yeah. And I just realized that I misspoke on the thymectomy piece, so I just wanted to correct that. So I said 6 months and it's actually 3 months. Okay, so it's less than 3 months from the time of screening. You have to be at least that far away. And you can't have a thymectomy that's planned during the course of the study either, because that could disrupt our interpretation of the results if there's, either information that's lost because you can't come to the study sites because of the thymectomy, and then the effect of the thymectomy itself that may happen on their MG.
Well, it does not look like I have any more questions on from our audience. So thank you so much, Dr. Guptill. We will, oh wait, I have one more. Will mask participants be in the same reporting cohort as double or triple seronegative?
I'm sorry, can you repeat it one more time, Dova?
Will mask participants be in the same reporting cohort as double or triple SNMG?
Yeah, so the study results will be on the overall population that's enrolled. Mhm.
Think I have now they're okay. No, that's okay. All right, we're good. Thank you so much, Dr. Guptill. We will be putting the webinar up for people to watch later, and we thank you very much for your time.
Great. Thank you all. Thanks
All
for
right.
participation.
Thank you. Bye-bye.